just say welcome to those that have joined the event already. We'll wait a minute or two to see if some others are still going to join us. We've had an excellent response to this webinar, so I'll give perhaps some stragglers some time to join us. So if we can just be patient for a minute or two. We are due to start on the hour at 2 o'clock. We'll see what the situation is then, and we can continue with the event. So thank you to those that have joined us already. So we'll just give it a minute or two for any others that still want to join in. Okay. So I can still see the numbers connecting into the webinar increasing here. So we'll just give it another minute or so, and then we'll start. Still got people joining. Okay, that seems to have stabilized. So I'll start this afternoon's event by welcoming everyone. I'm David Collins, the Chief Business Officer at Encardia. And although we're here to talk about science, I can't go past the title slide here without confirming a very exciting event in our company's history. The day before yesterday, we closed a significant round of investment, which will enable us to consolidate our scientific expertise and capacity and allow us to expand through the next year and into the following years. So our business plans are evolving, and we look forward to expanding upon the capabilities that we have at the moment. For those that don't know Encardia, there's just a very brief introduction here in terms of the main activities that we're involved with. So we can source stem cells, we can take human cells, typically fibroblasts or PBMCs, and reprogram them. We have our own differentiation processes for manufacturing human cells and working towards disease-relevant cells. One of the significant aspects of this is the scale that we can manufacture at, and supporting this for screening, we have assay development and disease modelling. The main area, or the original area of activity, was with cardiomyocytes, and we're well established now in the CNS neuronal area, and this is an expanding area of interest for us. And soon after I finish, we'll start talking about disease modelling capabilities and in-house assays. But in terms of using stem cell technology platform for drug discovery, we do believe that there are some significant benefits, because the cells and disease models can be derived from a single source at significant volumes, so we have consistency and reproducibility of the test system that's developed. Cells can be derived from patients, or genetic manipulation can lead to superior disease modelling, and we do believe that this human translational models really does improve the potential for success later in development and reduce attrition. So we do believe as an organisation that human stem cell technology does help bring better therapies to patients faster. What we're about to move into is the first in a series of webinars covering CNS-associated topics. So today we're going to hear about the manufacture and co-culturing of neuronal cell types. After this introductory session about the neuronal cell types, we'll move into using these in drug discovery, and then specifically in webinar 3, we'll look into neurodegenerative disease modelling. But it's now time for me to hand over to the speaker for today. Today's presenter is Isabella North. She obtained her PhD back in 2016 and subsequently worked in a postdoctoral role. And with that, combined with her time at Encardia, she's got extensive experience in modeling human neurodegenerative diseases with stem cell-derived neurons and developing functional assays. So I shall stop talking now, and gives me pleasure to hand over to Isabel to present today's webinar. Isabel.
Hi everyone, good afternoon. Thank you, David, for the kind introduction. So today I will be presenting the first webinar of a series dedicated to neuroscience and modeling of neurodegenerative diseases with the iPSCs. And I will be talking about today about the manufacture and co-culture of human iPSC-derived neuronal cells. So during this webinar, I will be talking about the benefits and the challenges of using the iPSC-derived neuronal models in drug discovery, particularly their large-scale manufacture, the need for reproducibility, and the importance of deriving functional and mature full differentiated cells. We will also gain some insights in the development of our differentiation in neuronal cell types and how to establish co-cultures. And we will go over two examples, the co-culture of cortical neurons and astrocytes, and the co-culture neurons and microglia. And also very important, we'll have some space for your questions. So we all acknowledge that it's difficult to develop translational in vitro models for the central nervous system. Historically, in the field, most of the work developed was performed in primary cultures and organotypic slices from rodent cultures, both in 2D and 3D models. With the availability of iPSCs, so human-derived induced pluripotent stem cells, we are now close to develop more physiological relevant human in vitro models. And how do we do this? So we try to be as close as possible in vitro with the events occurring in vivo. So we work very closely to the neurogenesis to obtain and culture structures similar to the neurotube and the neuroectoderm, and then we do the region patterning, so activating or downregulating canonical pathways as the sonic hedgehog, the Wnt signaling, or the BMP. But always with the idea to be as close as possible to the brain regions and the neuronal populations in an adult human brain. At Encardia, what we do is to combine the robust scale-up and at the same time maintaining the cell characteristics, so the identity and the function. So this is critical, and it's something that I will be talking about in the next slides. But all these process relies on the reproducibility. So we need to identify and control the so-called critical process parameters. So has the seeding densities of the cells, the scheme of the refreshings, the timing for the harvest, and also the continuous assessment of the critical quality attributes, the yield of the cells, the viability, and again the identity and the function of the cells. So today I will be talking about our CNS neurons, the astrocytes and cortical neurons, and with a special guest from the immune system, the microglia, that also plays a very important role in the physiology and pathology in the central nervous system. So I will start with our ENSITE CNS. ENSITE CNS is a mixed population of iPSC-derived neurons and astrocytes, as you can see for the expression of MAP2, a neuronal marker, and GFAP, a marker for astrocytes. We identify different subpopulations of neurons as the GABAergic neurons, so positive for GABA, dopaminergic neurons, positive for TH, and also glutamatergic neurons, positive for PGLUT1. As I said before, it's important to identify the critical parameters and also the quality attributes. In this case, we look at the levels of beta-tubulin, and you can see that for several batches, we have consistently high levels of beta-tubulin. When we look or qualify our cells at day 14 post-sowing, we identify a small population of proliferative progenitor cells, so they are positive for Ki67. Most of the population is neuronal, so they are battery-free tubulin cells, and at least half of the population of the astrocytes express S100 beta, that is a very or a classic marker for mature astrocytes. Regarding the functionality of this culture, so it's important to keep in mind that we have excitatory neurons and inhibitory neurons. So it enables us to qualify the firing properties of the culture on MEA at day 14. So what is important here is to look at the time in culture and to identify what is the best time window where we have the synchronicity and the organization in the network bursts in the culture, as you can see with these pink rectangles. And this balance of excitation and inhibition is very important for disease modeling, so in the case of epilepsy or seizure, and also for drug testing. Our next neuronal cell model is end-site cortical neurons. So it's basically a pure population of cortical neurons, so FOXG1 positive population, and more than 50% of those cortical neurons are committed to layer 5, and they are positive for TBR1 and CTIP2 markers. One aspect of this culture is mostly glutamatergic. We have a high activity on MEA even in very early stages of the culture, as day 4, and this makes this population very interesting to make a co-culture with astrocytes. Our astrocytes, they are also almost pure population of astrocytes, positive for GFAP. As you can see also here, one of the critical parameters that we look is the levels or the consistent high levels of GFAP for several batches. We have at day 10 post-sowing more than 85% of the population being GFAP positive, and more than 40% are S100 beta positive astrocytes. So when I was considering the cortical neurons to be a relevant population to be in co-culture with the astrocytes, it's because we know that the excess of glutamate in the culture is related with the excitotoxicity, and one way of balance these levels of glutamate in the culture is to add astrocytes. So astrocytes, they provide support to neuronal cultures through different roles, but one of those is the uptake of the excess of glutamate from the culture. And as you can see from the raster plot on your left side, we have a lot of activity, but it's very disorganized. When we had our astrocytes, we have the organization of the culture in the so-called network burst. So you can identify those with the pink lines, and this is a hallmark for a mature culture. So this reduction of the glutamate in the medium helps the emergence of these stable network bursts, and it helps to extend the culture time of the cortical neurons up to 4 weeks post-sowing. Also, our astrocytes, they play a role in neuroinflammation. So when we treat our astrocytes with a pro-inflammatory cytokine cocktail, IL-1 beta, interferon gamma, and TNF-alpha, we are able to measure in the supernatrants of the cultures several cytokines, as IL-6, IL-8, CCL2, and also the growth factor BDNF. Those are classical molecules related to the activation of inflammatory pathways. So we believe that the astrocytes will play a key role in the neurodegenerative diseases as Alzheimer's disease and Parkinson's disease. I took this image or this diagram from an application note that we put together with the CISBio for the validation of the HRF assay. My final cellular model is microglia. So we derive our microglia from monocytes enriched in the CD11b, CD14, and CD45. After 14 days in culture, we can identify the two phenotypes of microglia. So we have the ramified and amoeboid microglia that corresponds to the resting and the activated state of microglia. We are able to culture microglia with the M-site CNS, so there is a very nice interplay between neurons, astrocytes, and microglia, and has function when we activate or when we challenge microglia with the LPS, and in the presence of these green zymosan bioparticles or beads that are pH sensitive, we are able to see an increase in the number of green cells, which means that they were able to do the phagocytosis of these beads. So in summary, I hope that we got this message that we have these building blocks or these cell types as the neurons, as the astrocytes, the microglia that are characterized for the identity, for the functionality, and they are able to play important roles in neuroinflammation, in the crosstalk between neuronal, glial, and immune cells. And this is particularly important in the neurodegeneration and other diseases. And as examples, Parkinson's disease, Alzheimer's, frontotemporal dementia, and others that have this component. In summary, I hope that as take-home message, we believe that the robust manufacturing processes and appropriate assays, both during process and also in release testing, are critical for reliable identity and functionality of the neuronal cell types. And the platforms that we derive from iPSCs, both the neurons or co-cultures, are physiological relevant in human in vitro models. And we'll be talking in more detail about the assays that we are able to run and the kind of diseases and phenotypes that we can study using this technology. So from my side, this is my last slide. And then I will be happy to take your questions. Thank you for your time, your attention.
Thank you very much, Isabel. It was very concise. We're beginning to get some questions through here, and I would encourage anyone to continue typing. I've got one question here, Ray. S100B and the organized EFIS data, is there any indication that the S100B plus cells are acting as oligodendrocyte-like cells? Can you see that in the chat, Isabel?
Okay, now I think I can. Yes, now I can see. Okay, no, we don't use that marker for oligodendrocytes. We don't believe that we have oligodendrocytes in our M-site CNS model. We use, or I forgot to say that in the slide, but we use to characterize our GFAP population with S100B and also aquaporin to identify the mature part of the population of astrocytes. So it's more focused on the level of maturation of astrocytes rather than to identify oligodendrocytes.
And regarding astrocytes, Isabel, what is the percentage of astrocytes in the CNS population? Do we know, and why? So
we have a 30 to 40% percentage of astrocytes in our M-site CNS. And in the slide, I think I presented that we have at least 50% of that population to be mature, because it's something that we try to link the identity and the functionality of the astrocytes. So the more or the most functional astrocytes are the mature ones.
Okay, thank you. Got some questions. If there's any further ones, then please do type them in and send them through. This one's perhaps preempting the future webinars, Isabel. But can we say a few words already about how we would model Parkinson's disease?
Yes. So Okay, so about the Parkinson model. So we know that when we think about Parkinson's disease, we relate with the substance and dopaminergic neurons, but there have been several studies also linking the cortical neurons and the involvement of the cortex in the dimension component of the disease. So we could offer cortical neurons to study that component, for instance.
Okay. We've got some more questions coming through. For the mixed cultures, are the cells frozen down together or separately?
They are frozen together. So I think it's one of the most important thing about this culture is ready to use. So basically, we just have to wait for a particular time point to make our essays. Okay. So no premix.
And presumably a benefit of that is if you've manufactured a big batch, then you've got a consistent mix ratio throughout the whole batch as you use it.
Exactly.
Yeah. We've got more questions coming through. This is great. Thank you to everyone that's sending them in. Do we have any data using the InUsight live cell imaging?
So for microglia, we did some live cell imaging, but it was not using the InQsight equipment. So it was a similar one. I believe that some of our essays we have some live cell imaging. So I think in the next webinars we'll be talking about these essays. From this webinar, the uptake or the phagocytosis assay was done with the live imaging.
Thank you, Isabel. And this is probably going to lead in again to the disease modelling and the webinars that we've got planned early in the new year. But how often do we work with CRISPR-edited stem cell lines?
So I think it depends on the cell type. If they are in cardiac cells, I believe that we have more projects ongoing. Right now, I think in the context of disease modeling, we don't have any line yet, or we are not working currently with any line.
Well, I think that's the questions have slowed down in terms of them coming in, and we've answered all those. So thank you very much to everyone that's submitted a question. What I think we can bring this webinar and the discussion to a close here. If you are interested in extra information, I've just opened up access to a paper that can be downloaded. So you're welcome to take a copy of this, and there is contact information on the last slide here should you want further discussion with the scientists involved in this work. So at that stage, I'd just like to thank everyone that took time out of their busy days, no doubt, to connect with us and listen to this webinar. And hopefully you'll join us in the new year when the series continues. So thank you very much, everyone, for your participation. Thank you, Isabel. And I say hopefully we'll see everyone in the new year after a great, I can't believe that I'm talking about a Christmas break, but we're entering that time of year. So thank you, everyone, and see you in the new year, hopefully, if not sooner, if you've got questions. Thank you.
Bye.
And I'll close down the meeting in a minute or two to give people time to download the poster. So thank you very much.